So it's actually a good transition towards the next presentation that will discuss learned parcellations and their dependence on the data you put in, so with a state-specific parcellation of the human brain.
Right, thank you. Following up on what Sal and Sarah just shared with you about the different parcellations which reveal different insights about the underlying properties of the brain, today I would like to talk a little bit about some of our recent works on individualized and state-specific parcellations. To give you some historical overview, so for so long, neuroscientists have been interested in identifying brain atlases by dividing the brain into different regions and understanding the properties and the functional role of these different regions. And perhaps one of the very early attempts to identify an atlas dates back to 1909, when Brodmann divided the brain into 52 regions based on the cell type or site architecture. And since then, there have been many other attempts based on the brain anatomy and morphology, and also more recently based on brain function. So what it means is that, for example, you would acquire some functional magnetic resonance imaging or different functional data from the brain, and based on the temporal synchrony of different regions, you would basically group regions together and define atlases. But based on the particular property that you choose, you would end up getting different types of atlases. And even within a particular property, different methods, different data would generate different types of parcellations. And although no parcellation or no atlas so far has emerged as the dominant one, there's this assumption that such an atlas exists, and we would like to ultimately get closer and closer to this, to approximate this atlas by acquiring more data or having more fancier methods and more advanced technologies. And while traditional approaches have been mainly focused on basically dividing these, generating parcellations or atlases at the group level, we would like to ultimately move on to single subject level, where instead of averaging data over all the population, we preserve these subtle differences across individuals. And there have been amazing works recently on individualized atlases and individualized parcellations, and how the differences in these parcellations can reveal the differences in different individual phenotypes, basically. But what I would like to focus on today mostly is more about, even within a single individual, across different states, the functional organization of the brain is changing. So we would expect that the functional atlases or the functional parcellations would also change based on the underlying cognitive state of the brain. So whether you're watching a movie or whether you're listening to music, engaging different types of tasks, you would expect the parcellation of the brain also change to reflect such states, basically. So today I would like to answer this question, that whether there's a single parcellation, single universal parcellation for all cognitive states, or whether the parcellation of the human brain changes reliably to reflect the underlying cognitive state. To answer this question, we focused on single subject analysis first, because we wanted to make sure that our results are not going to be confounded by individual differences in function anatomy. So we acquired data from a single subject, in this case, my advisor, Todd Constable, and we acquired 30 sessions of fMRI. Within each session, the subject was doing 6 different tasks and 2 different resting states. And this whole process, this whole task design and pre-processing is basically done by my great collaborator of mine, Abigail Gray. So we acquired these fMRI sessions, and we developed an individualized, an exemplar-based individualized algorithm, where we start from a group parcellation, and we define exemplars within each of the nodes that are defined by the group. And these exemplars are basically the voxels that are going to best represent each of these nodes. And then after identifying these exemplars, we just remove or eliminate those boundaries that are already defined by the group, and we assign voxels to the closest exemplars. So this way we come up with this individualized atlas that best reflects the underlying data. And we assign these voxels to the exemplars in a way that we also preserve spatial contiguity in this case. And I'm not going to go to the details of the algorithm, but I would definitely be happy to speak about this offline. And also, please check out the paper if you're interested. All right, so we wanted to see whether the parcellations that we created are more similar or like more reliable within a state and are changing across different states. So to answer this question, we took advantage of statistical ensemble analysis, where after we created these parcellations for each of the 30 sessions, we divided these 30 sessions into 2 equal-sized groups, each of them having 15 sessions. And then we created the majority vote of those groups. So for every state or task, here I would basically alternate between state and task, and what I mean is a task-induced cognitive state of the brain. So for each of these tasks or each of these states, we have 2 different parcellations. And note that these parcellations are coming from non-overlapping sessions, so we're not really dealing with the session effects here. And given that we're interested in seeing how similar these parcellations are across different states and also across different, and also within a single state, we generated a similarity measure, like a pairwise similarity matrix between all these parcellations. So we came up with this m by n similarity map, where each element, say for example element 1 and 3, is basically telling us how similar parcellations are between state 1 and state 3. We repeated this entire analysis 1,000 times to generate a distribution of parcellation similarities, both across different states and also within a single state. All right, so in the previous map, just note that this similarity map is actually showing, for example, on the diagonal, you're showing the parcellations within a single state, how similar the parcellations are within a single state, and on the diagonal elements are showing the similarities across different states. So to show some results, we observed that the parcellations are actually significantly more similar on diagonal, which means within a single state, comparing to the off-diagonal, or across different states. And we used 2 different similarity measures to basically assess both local and also global properties of the similarities. On the left, I'm showing the Hamming distance similarity, which basically is kind of counting the number of voxels that are changing their association across different states and also within. And on the right, I'm showing the node size correlations. And this is basically the average of all of those 1,000 matrices, and here I'm showing the distribution. So the blue histogram is showing the distributions of cross-state similarity, and on the orange you can see the distributions of within-state similarity. So as you can see, there's no overlap between the 2. Like the 2 distributions are significantly different, with within-a-state parcellation similarities being significantly higher than cross-state parcellation similarity. So far, we've kind of observed that the parcellations are more similar within each state and are different across different states. But we wanted to see whether these parcellations or whether these reconfigurations are reliable, such that we can predict the underlying state just by using the parcellation maps. So to answer this question, we did some cross-validated analysis where we trained and tested a gradient listing machine, or like a classifier, using node size vector as a feature, which is perhaps one of the very coarsest measures of parcellation. And by node size, I'm referring to the voxels that are assigned to each node. And the answer is yes, we can significantly predict the state just by using the parcellations. And it means that the parcellations are reliable enough within the state to kind of form a signature of that state. And there was another interesting observation. We saw that even within one state, the parcellations are obviously not identical across sessions. There are some subtle differences between the parcellations, even within one state. And we wanted to see if these reconfigurations are meaningful or reflecting something about the underlying cognitive state, or are they just random? So to answer this question, we tried to predict how well the subject was doing the task in the scanner, based on the parcellation maps, again, just using the node size vectors. And you could see that we can significantly predict how well the subject is doing the task, only by using these coarse features, which is just a node size vector, which means that the parcellations are really changing in order to reflect the underlying state, such that they can reveal information about the state of the brain at the time, how well the person was engaged in the task during the performance of the task. And to make sure that these observations are not specific to Todd's brain, we replicated all the analysis using a publicly great, amazing work, publicly available data set and Midnight Scan Club, which consists of 10 subjects. Each of these subjects are scanned during 10 sessions while doing 5 different tasks and also harnessing state. And interestingly, we observed that all observations are basically still holding in this situation, where the parcellations again are more similar within one state and more different across different states. And we can again significantly predict the parcellations just by using the node size vectors. So far, all the results that I have shown you are mainly focusing on single individual and how the parcellations of a single individual is changing across different sessions and states. But then we wanted to see whether the same property holds when we consider the parcellations across different individuals. To do this analysis, we took advantage of the publicly available dataset Human Connector project, which I'm sure you're all aware of. So we used basically 514 subjects after filtering for motion, and these subjects were engaged in 7 different tasks in 2 different resting states. And again, we could see that the same results are observed, meaning that the parcellations are more similar within a single state, even across different individuals. And we can, again, significantly predict the brain state for a novel subject, for a subject that we haven't seen, just by using the parcellation maps. So again, this means that the parcellations are really state-dependent and are changing to reflect the underlying cognitive processes. So to summarize, hopefully I've been able to show that the parcellation of human brain is state-dependent, and these state-evoked reconfiguration of nodes are functionally relevant as they reflect the underlying cognitive state of the brain, whether it's a task or whether it's like a task-induced state or whether it's based on the task engagement during a specific state. And basically, parcellations can give us a new metric for assessing the cognitive states, and we would also hope that these results are useful for functional connectivity approaches that are taking to understand the differences in functional connectivity patterns across different states. With that, I would like to thank my colleagues and collaborators and also announce that all the codes are publicly available in our GitHub repository and also part of our open source software, Bioimage Suite. So please feel free to check them out and play with them. And with that, I would like